Dear students, today's topic is study on diseases of onion and guava. First of all, diseases of onion. Onion is the most important bulb crop in India. It is grown well in many soil types and is grown extensively in many parts of India. A sizable quantity of onion bulbs are exported from India to various countries. Several factors have been identified for low productivity. The most important factors responsible for low productivity are due to fungal diseases. Important fungal diseases. First one is the onion smudge. The symptoms appear as dark green to black spots, blackening with age, and sometimes scattered over the surface of the bulb, but are more frequently concentrated in smudge-like spots, roughly circular or arranged in concentric rings of 1 to 2 cm in diameter or more. Next is the scientific classification. Kingdom fungi, phylum Escomycota, subphylum Pagizomycotina, class Sorderomycetes, order Glomerellus, family Glomerullaceae, genus Coletotrichum, species Sarsinans. Now coming to the character of this pathogen. Disease is caused by Coletotrichum sarsinans. Acervuli are formed on stomata just beneath the cuticle by formation of pellicid layer of short conidiophores along with dark setae. Conidia are fusoid, falcate and hyaline. Next disease is the purple blotch. This disease is common in India both in seed as well as in bulb crop and mostly aggravated by botrytis infection and thrips attack. Disease appear as small water-soaked lesions on the leaves or seed stalks that quickly develop white centers, become zonated and brown to purple. The diseased leaf or stalk cause drooping. The infected plants fail to develop bulbs. Next is the scientific classification of this pathogen. Kingdom is fungi, phylum Escomycota, subdivision Pagizomycotina, class Dotiriomycetes, order Pleosporales, family Pleosporaceae, genus Alternaria. Next is the pathogen character. The fungus responsible for this disease is Alternaria pori. Conidiophores arise singly or in groups and are straight or flexus, often geniculate, septate, pale to mid brown in color. Now coming to the conidia. Conidia are muriform or curved, obclavate and taper to a beak that is commonly about the same length or slightly larger than the body of the conidium. Each conidium has 8 to 12 transverse and 0 to several longitudinal or oblique septa. The beak is flexus, pale and tapering. Now coming to the next important disease, that is the downy mildew. This pathogen can be examined when infected plants show circular to elongate lesions and infected stalks break over with the weight of the seed umbel, thereby causing the seed to shrivel. The fungus can be observed as a violet growth, later become pale greenish yellow, and ultimately the leaves or seed stalks collapse. Now coming to the scientific classification of this pathogen. Kingdom is Chromalveolata, phylum Heterocontophyta, class Omycota, order Peronosporales, family Peronosporaceae, genus Peronospora, and species is Destructor. This is about the scientific classification of this pathogen. Next is the character of this pathogen. The disease is caused by Peronospora destructor. The mycelium of the fungus is non-septate, intercellular with filamentous haustoria. The sporangiophores are aseptate, hyaline, swollen at the base and 3 to 4 times dichotomously branched. The stigmata are acute to subacute and bear pyriform to fusiform sporangia. Oospores develop late in the season and germinate by germ tube. So, this is about the character of this pathogen. Next disease is the white rot. The fungus invades roots and the basal part of the bulb scales, appear as yellowing at initial stage and wilting of the leaves followed by a total collapse of the tops. The bulbs become soft and water soaked. White fluffy cottony growth of mycelium can be observed. Black sclerotia resembling mustard grain can be seen on the infected bulbs. Now, let us see about the scientific classification of this pathogen. Kingdom is fungi, division Ascomycota, class Ascomycetes, order Haloceales, family Sclerociniaceae, genus Sclerotium, and species Sepivorum. Now, coming to the character of this pathogen. The disease is incited by Sclerotium sepivorum. The fungus produces sclerotia are black, globular, which are uniformly round, measuring 0.35 to 0.5 millimeter in size, which appear on the fluffy white mold, and sometimes larger black sclerotia crusts also form. Sclerotia germinate only once. Next disease is the stem phylum blight. Symptom expressed as small yellowish to orange flecks or streaks in the middle of the leaves, later develop into elongated spindle shaped spots which is surrounded by pinkish margin. It appears on the inflorescence stalk which damages to seed crop. Now, coming to the scientific classification of this pathogen. Kingdom fungi, phylum Ascomycota, class Dotidiomacetes, order Peronosporales, family Pleiosporaceae, genus Stem phylum, species Vesicarium. Now coming to the pathogen character. The disease is caused by stem phylum Vesicarium. Conidiophores are straight to variously curved 1 to 4 septa, simple or occasionally one branched. Conidia are oblong or broadly oval and sometimes in equatorial 1 to 6 transverse septa and 1 to 3 longitudinal septa and constricted at the major transverse septa. Conidia are light to medium golden brown to olive brown in color and each has a conspicuous basal scar-like zone. SI are cylindrical to clavate in shape. Ascospores are ellipsoidal and the upper half is narrowly tapered. Next is the diseases of guava. First is fungal diseases. Name of the disease is the Fusarium wilt. The disease is characterized by light yellow foliage with loss of turgidity, epinacity, premature shedding of leaves and defoliation. The infected parts fail to bring new leaves, flowers and eventually dry up. Fruits remain underdeveloped, hard and stony, and later the entire plant defoliated and plant dies. Next is the scientific classification of this pathogen. Kingdom is fungi, division Ascomycota, class Sorderomycetes, order Hypocreals, family Nectriaceae, genus Fusarium and species is Oxysporum. Now coming to the pathogen character. Causal organism is Fusarium oxysporum, forma specialis CD. Mycelium is white or pink with a purple tinge. Microconidia are oval to ellipsoid, cylindrical, straight to curve and 7 to 10 into 2 to 3 micrometer. Macroconidia are 3 to 4 septate and 32 to 50 into 3 to 7 micrometer in size. They are fusoid and pointed at both ends. Chlamydospores may be intercalary or terminal. This is about the character of this pathogen. Next disease is the anthracnose or dieback or fruit spot or twig blight. The infected young shoots, leaves and fruits are readily attached while plants are tender. The greenish color of the growing tip changes to dark brown and later turn black necrotic, extending backwards. Now let us see about the scientific classification of this pathogen. Kingdom is fungi, division Ascomycota, class Sorderomycetes, order Glomerulales, family Glomerulaceae, genus Coletrotrichum. Now coming to the character of this pathogen. Pathogen is Coletrotrichum gliosporoides. Conidia are hyaline, aseptate, oval to elliptical or straight, cylindrical or flattened at base. Conidiophores is cylindrical and tapers towards apex. It is hyaline and septate with single terminal phyllite. Acervuli are dark brown to black. This is about the character of this pathogen. Next disease is the stem canker. The pathogen which mainly attack on stem branches causing crack lesions. In fruits, symptom expressed as light brown spots at stalk. Later, the entire fruits become dark brown to black and mummified, resulting in dieback symptoms. Now let us see about the scientific classification of this pathogen. Kingdom is fungi, division Ascomycota, class Ascomycetes, order Gilariales, family Hyponectriaceae, genus Physiolospora, species CD. Now coming to the pathogen character. The fungal perithecia are small brown to black structures which develop at the infected stem. Perithecia are glabrous with a fleshy wall. Ascospores are hyaline, narrow, ellipsoid and one-celled. Conidia are single-celled, ovoid with a rough wall and measure 20 to 26 into 9 to 12 micrometer. On the stems and fruits, pycnidia are formed in stroma. So, this is about the character of this pathogen. Now, coming to the conclusion of this topic. There are several diseases observed on onion and guava caused by fungi. The initial identification of the disease can be obtained by symptoms produced by the pathogen to their host, and later the confirmation is obtained by the presence of the pathogen in infected area. The management of the diseases depends on proper identification of the disease, pathogen involved and characters of the pathogen.